Hi kids, are we live? Hello, are we live? Good morning everyone. Can you see me, hear me? Can you see me, hear me? Good morning, good morning, good morning. Welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and today we are going to discuss the embryonic development in detail. So we will start from the first step, that is fertilization, and then different stages of development about the placenta, hormones release, even the parturition and the lactation, right? Even the parturition and the lactation. I hope I'm clearly audible to you all. I have seen my last lecture, right? The voice was not so clear, bache. Today, I hope it is. Clear? Done? So many of you were asking about the last part of the human reproduction. So in this way, we are going to complete it. And tomorrow at 9 a.m., we are going to take another session, that is the reproductive health, and it will be in the one short way. Clear? Done? So tell me what you have studied so far. Tell me what you have studied so far. See, I will not teach you as per the test series, right? Because you know that, bache, in that test series, it is for your revision. Okay, so but in the evening, because we have already completed the cell cycle in one shot, so yes, I'm planning to start the biomolecules. So one way or another way, we are going to cover it, and all the topics of the diversity, right, that is living world, biological classification, plant kingdom and animal kingdom is done, right? But guys, you know, the live learners, they are quite less, their number is quite less, so I don't think that you need these sessions in the morning. Do you need that? Huh? And quickly hit the like button, and thank you so much for making us 60K subscriber family. Increase the number, kids. Increase the number. Yes, tell me, do you want these sessions in the morning? Ma'am, I want to join the NEET Energized batch. So, bache, Harsha, just go to the description box. There is a link. Click there, add your details. You'll be the part. Do you want the morning lectures? Telegram link for the test series will be provided to you. Anything else? Anything else? Anything else? So guys, quickly, how many of you are interested in these morning sessions? Tell me quickly, otherwise we'll take the evening sessions only. Quick. Otherwise, I'll take the evening sessions only. Okay, so this is what we are going to start. That is the embryonic development. Okay, that is the embryonic development. Okay, so I'll ask you a few questions and answer them, obviously. So you know that already what we have covered so far. When you talk about the human reproduction, I told you some series of events are there when we talk about the reproduction, isn't it? There are some series of events that we discuss when we talk about the human reproduction, and I have given you the trick for that, and that is give, sip and go. Do you remember? Do you remember this trick? That is give, sip and go. This is what we have already, right? This is what we have already discussed and the already discussed, right, bache? So when you talk about this give, G stands for gametogenesis. What is it? G stands for gametogenesis. I stands for insemination. F stands for fertilization, right? C stands for cleavage, and then this I stands for implantation, right? I stands for implantation. P stands for placentation. Note down these steps, bache. Then comes the gastrulation. then the organogenesis, that is organ formation, and finally there will be the parturition, isn't it? There will be the parturition. This is what we have studied so far. So it is give, sip and go. What is it? It is give, sip and go. So gametogenesis, right bache? Gametogenesis and insemination, fertilization, cleavage, implantation, placentation, gastrulation, organogenesis, and finally there is the parturition. So this is what we have already covered. When you talk about the genesis means formation, gametogenesis means gamete formation, and here we talk about the spermatogenesis and the oogenesis. That is what we discuss, right? Spermatogenesis and the oogenesis. Then comes insemination. The male gametes will be transferred in the female genital tract, right? Ejaculation of semen will be there in the female genital tract during the coitus, during the copulation. Then comes the fertilization, right? So after fertilization, what is going to happen, bache? After fertilization, there will be the formation of zygote, and that zygote will undergo these rapid mitotic division, that is the cleavage. So after cleavage, the further development will be there, the implantation, right, placentation, gastrula formation, organ formation, and finally the childbirth will be there. Okay, so today what are we going to study? Today we will talk about this part, that is fertilization. Okay, firstly we will start from this part, that is fertilization. Okay, acha, tell me one thing, do you know the difference in between the word ovum and the egg? Do you know the difference in between the word ovum and the egg? Gracie, ma'am, why the test is not conducted till now? Which test are you talking about, Gracie? Tell me, what is the difference? Gracie, you please type your concern here, and other students, please tell me, do you know the difference in the word ovum and the egg? Anyone? And quick, please be quick. Anyone? What is the difference in the word egg and the yes, and the ovum? Ovum is before fertilization. Okay, anything else? Anything else? Quick, anything else? Egg after fertilization. No, it's not like that. See, when you talk about the egg, right, when the word is when the word is egg, egg means your ovum or oocyte, right? It can be your ovum plus oocyte, and then comes the egg membranes. So we are going to start from this part, okay? And then comes the egg membrane. So we are going to start from this particular part. So see, try to understand it. You know that after fertilization, we have already covered the menstrual cycle. So you know that on the 14th day, right, when the menstrual cycle is of 28 days, so on an average on day 14th the ovum will be released. Actually it is the oocyte, it is not the ovum, it is basically the oocyte. It will be released, yes or no? Guys, be quick, right? You need to answer the whatever I'm asking, just answer it in the chat section and be quick. Okay, so what is going to happen on the day 14th? The ovum will be released, the egg will be released, isn't it? That's the fact, isn't it? Yes or no? It will be released. Now you know that that is basically an oocyte. Okay, that is basically an oocyte which will be released on the day 14th of the menstrual cycle. And today you just need to listen to me like I'm telling you a story. Okay, in a story format you need to understand everything. Do not relate it with your NCERT right now. Do not think about any exam, right? Just be curious that today you are going to learn something related to the human body. Okay, right? This should be your agenda. Today we are going to learn something related to what? Related to the human body, right? And this is what you guys are going to discuss in the MBBS as well. So you should feel good that, okay, we'll get to know about the human body, how we all are developing. Fine. So oocyte will be released, and you know that oocyte is having the metaphase to arrest, isn't it? That oocyte is having the metaphase to arrest, isn't it? Now you know that this oocyte is without centriole, and the nucleus is having the metaphase to arrest. Now that egg will be released, you know that near to the ovaries, let's say if this is the ovary, right here you will see some finger like structures. You guys are going to see some finger like structures here, yes or no? This is what you people will see. We know about the structure of the female reproductive system. So you know that the closest part of fallopian tube to the ovary is your infundibulum, and that infundibulum is forming some finger like structures which are known as fimbriae, and that fimbriae will pick up the egg from the ovary. And when this egg will pass from here, right, this egg, actually it is the oocyte, so it is, you know that it is having its own plasma membrane. It is having its own plasma membrane. It is also covered with a vitelline layer. Do you remember? It is also covered with a vitelline layer, right? And then in its surrounding, you will see another layer, and that is known as, right, that is known as corona radiata. Do you remember? Right, in the last lecture, bache, when we were discussing the oogenesis and menstrual cycle in detail, I told you about all the layers of this particular egg, right? I told you about the layers here, isn't it? I told you about the layers here. So you know that we have the plasma membrane inside, and this one green in color is zona pellucida. What is it? It is the zona pellucida, which is acellular, which is not made up of any cell. It is just made up of glycoproteins, right? And then in its surrounding, this layer is your corona radiata. Right, it is corona radiata, and that corona radiata is formed by the ovary. Like even in the paper, this is what they can ask you, bache. This zona pellucida is your primary egg membrane. What is it? It is your primary egg membrane. Fine, it is your primary egg membrane, and it is formed by oocyte itself. This oocyte is going to form it. So it is going to form by oocyte itself. And when you talk about this corona radiata, it is secondary egg membrane, right? It is secondary egg membrane, and it is formed by the ovaries. Clear, bache? It is formed by the ovaries. Right? And here in the surrounding, because these ovarian cells are forming it, so definitely it is cellular, right? Ovary is forming it, right? But it is made up of ovarian cell. Now corona radiata is actually the innermost layer. This surrounding, in the surrounding you will see the cumulus here. What is it? It is the cumulus. So actually when this oocyte will be released from this part, no, this oocyte, when it is covered with the egg membranes, you are going to call it as egg, right? When this oocyte, it is covered with the egg membranes, then at that time we are going to call it as what? We are going to call it as egg. So if it is simply oocyte, you are not talking about the egg membranes. You are not talking about the primary and the secondary membrane. So if it is the egg, we are talking about these membranes as well. So I hope it is clear. Zona pellucida, it is formed by the oocyte itself. It is the primary egg membrane. Corona radiata is formed by the ovary, and it is the secondary egg membrane. And if you will see here in the surrounding, this is the cumulus. What is it? Cumulus, made up of ovarian cell. So we also say that corona radiata is the innermost layer of the cumulus. Clear, bache? It is the innermost layer of the cumulus. Clear, bache? So now you know that this gamete will come here, right? The ciliated epithelium of the fallopian tube is going to push it, right? It is going to help in the movement of this ovum, yes or no? Yes or no? Now you know that in this fallopian tube, there will be one part that is known as ampulla. Remember, kids? There will be ampulla, right? Ampulla. And you know that it is the widest part of the fallopian tube, and it is the site of fertilization. So if the sperm is present here in ampulla, it is going to fertilize the ovum. Okay, it is going to fertilize the ovum. Is that clear? Fertilization is going to take place here. Done, bache? Any doubt? Any doubt? So if there is the unprotected sexual intercourse, if semen is present in the female vagina, these sperms are going to travel towards the fallopian tube. So if at the same time, if egg is also released and that egg is present in the ampulla and even the sperm is present in the ampulla, so it will result in the fertilization of two and the zygote formation will be there. And if there is no sperm, after 24 hours, the ovum will be degenerated. Okay, the ovum will be degenerated. Are you getting it? Are you getting it? The ovum will be degenerated then. Clear, bache? So this egg, this ovum, it is having the fertilizing capacity for the initial 15 hours of its release, right? It will get released from the ovaries for initial 15 hours. It is having that fertilizing capacity. It is having that fertilizing capacity for initial 15 hours. Even after that, even that fertilizing capacity will also decrease. Okay, okay. So within 24 hours, if it is going to get that sperm, fertilization will occur. Otherwise, there will be no fertilization. Any doubt? Any doubt? Right? So obviously, after this stage only, there will be the embryonic development. We need to form the zygote first and then we have to study the embryonic development. So that is how we are going to proceed, bache. So here you people can see this diagram, right? So see the egg, the ovum will be released, right? So it will be fertilized, right? And further divisions will occur. The blastocyst will be implanted here in the uterus. Okay, it will be implanted here in the uterus. So this is what we need to discuss now, right? This is what we need to understand now, and then I will show you this particular diagram. So before understanding anything, we need to know about one more thing, and that is the capacitation, right? But the topic of capacitation is not, given in the NCERT, but capacitation is important, right? So today, what are we going to discuss first? We are going to talk about the capacitation of sperm in the case of humans. Okay, let me tell you, spermatozoa is singular, sperm is plural. Okay, spermatozoa is singular, sperm is plural. Clear? Clear? So what is this capacitation? Do you know? Do you know that? What is the meaning of capacitation? Anyone in the class here? What is the meaning of capacitation? We'll start with the capacitation and then we'll talk about the next step. The changes occurs in the sperm, I think. Seriously? Yeah, give me the direct answer. Yeah, what is this? I think. Nahi, start revising properly, na? Start revising properly. Tell me. Okay, I'll ask you one question. So on an average, in a healthy individual, what amount of semen will be released per ejaculation? Anyone in the class? Right? During sexual intercourse, during coitus, right? Obviously, the semen will be ejaculated by male in the female genital tract. So, can you tell me the quantity of that semen? What is it? What is it? You know that, na? Their semen is there. In a healthy individual, there is a certain amount. The semen will be in that quantity. 200 to 300 what? 200 to 300 what? But tell me the units as well. 3 to 4 ml of, okay. So basically you know that 200 to 300 million of sperms, like I'm asking about that milliliter or something. Guys, you really need to check it. Let me tell you. You really need to start revising it. Right? I think you people are confusing it. If you remember, if sperms are less than 20 million, if less than 20 million sperms per ml is present, it is oligospermia. That is, the sperm amount is less. And per ejaculation, how many sperms are released on an average? Check it. Check it properly with the units and then let me know here in the chat section, right? That is your homework. So you know that the ejaculated sperm, out of them 60% should have that normal shape. They should have that vigorous motility, right? Like some of them should have proper shape, some of them should have the proper motility as well. We need it, right? We need it. So at least 60% of them should be very, very, very healthy, right? Now there is one process, that is the charging of sperm. The capacitation of sperm is also known as charging of sperm. What is it? It is the charging of sperm, right? So basically capacitation is the set of physiological activities. It is the set of physiological activities that will occur in sperm, in female genital tract, right? It is the set of physiological activities that will occur in sperm in female genital tract, right? In female genital tract. So it is very important. If the sperm is not charged, it is incompetent, it is not able to fertilize the ovum, right? This is what you need to remember. This is what you need to remember. So what is capacitation? The set of physiological changes, the set of physiological activities that will take place, right? That will take place in the sperm in female genital tract, and it will make sperm competent to fertilize ovum, right? Competent to fertilize ovum. So this is what you need to remember. Done, bache? This is what you need to remember. No capacitation, no charging. So you know, even do you know that incapacitated sperm, it will get trapped in cumulus itself. It is not able to cross the cumulus. Okay, it will be trapped in that cumulus. It cannot even cross that cumulus. You know that after cumulus, it needs to cross the corona radiata, right? And then it needs to, you know, join with the zona pellucida, right? So if it is incapacitated, so obviously it is incompetent, it will get trapped in the cumulus. This is what you need to keep in your head. Clear, bache? Clear, bache? So now what type of changes are going to be there in this capacitated sperm? Can you tell me about that changes? Can you tell me about that changes? And let me write. Okay, so even note down this thing, bache. You tell me about the changes and then note down these points. Note down these points. Capacitation is not given in NCERT, but it is important. It is important. Okay? It is important. Yes. So, see, for the capacitation, calcium ions are required, bicarbonate ions are required, and then serum albumin is required. See, when you talk about the sperm structure, you know one thing very clearly, right? When you talk about the So when you talk about the sperm structure, you know that the head of the sperm will contain the acrosome vesicle, yes or no? You all are well aware of it? Yes, bache. You all are well aware of it, yes or no? If there is any doubt, please check the last session, okay? Please check the last session. So you know that here in the sperm, here you will find the acrosome. This is the nucleus here, right? And here you will see the centrioles, and that spirally coiled mitochondria will be there. Spirally coiled mitochondria will be there, which is Nebenken, and here you have the structure that is known as acrosome, right? And this, in the surrounding, we have the plasma membrane, isn't it? This is the plasma membrane of the sperm. Now, bache, this sperm, it is present in the semen. Obviously, it is covered with some compounds also. Right? It is covered with some compound also. It is not just the sperm which will be released in the female genital tract. It is actually the semen, right? It's a fluid where you will find the fructose as well, some, you know, clotting agents as well, right? And the enzymes are also there, right? The ions are also there. Prostaglandins, which are meant for the contraction of the genital tract, even they are present, right? So all the important things which are important for the nourishment of sperms, for the movement of sperms, they are present in that semen. Now when you talk about this sperm, about this plasma membrane, so what is going to happen here in this plasma membrane? This plasma membrane is also having the cholesterol. What is present in the plasma membrane of the sperm? Cholesterol is present. Now during capacitation, what is going to happen, bache? During capacitation, what is going to happen, bache? This removal of, right? This removal of cholesterol this removal of cholesterol from plasma membrane of sperm occurs. Okay. This cholesterol will be removed from the plasma membrane. This cholesterol will be removed from the plasma membrane and it result in high pH in sperm. Right? It will result in high pH in sperm. Slowly the pH here will increase. Are you getting it? pH here will increase. Right? So loss of cholesterol from this plasma membrane, right? Loss of cholesterol from this plasma membrane leads to the pH rise in sperm, high pH in sperms, right? Due to which even the sperm will also undergo the acrosomal reactions. I'll explain it, right? Due to which the sperm undergoes acrosomal reaction. Okay, the sperm undergoes the acrosomal reaction. The first point clear, bache? First point clear, right? And moreover, here you will find some proteins and carbohydrates which are covering the sperm structure here, right? Even here, what will you see? Some proteins and carbohydrates, they are also lost. They are basically blocking the recognition sites. Let's say if this is the first point. So the second point is removal of proteins or carbohydrates. Removal of proteins or carbohydrates from sperm structure. Okay. And these proteins and carbohydrates, they block the recognition sites. So here on the sperm structure, now you will find some, receptors which are going to bind with the egg membrane. So some proteins or carbohydrates, they are covering it. So during the capacitation of sperm, this is going to happen, bache, that proteins and carbohydrates will be removed, right? They will be removed, and then that the receptors will, right? That the receptors will come on the sperm surface. And do you know how will it occur? Yes, bache. Actually, what is happening in this sperm when there is the removal of this? You must be thinking that, ma'am, how such changes are going to take place? Because when there is the removal of cholesterol from plasma membrane of sperm, now I'm telling you that high pH will occur. Actually, your K positive ion, right? What is going to happen? What is going to happen here, bache? Your K positive ions will leave, right? K positive potassium ions, they will leave the sperm. During charging of sperm, the potassium ion, they will leave the sperm. So now you know that if potassium ions, they are leaving the sperm, the inside will become negative, yes or no? It's very basic, right? It's very basic. I'm going to relate it a bit with the neural control and coordination. Tell me about this. Yes or no? Yes or no? Yes or no? This is basically the third step, right? This is basically the third step. So all I'm saying is that, see here we have what? We have the sperm. So from this sperm, what is going to happen, bache? From this sperm, the K positive ions, they will leave the sperm. So if K positive ions, they are leaving the sperm, it will change membrane permeability. It is going to change membrane permeability, right? Because inside will become negative now. Right, inside will become negative now. So due to which what will happen? The calcium channels, they will open. Right, it is the calcium which is very important here. During fertilization, right, during fertilization, calcium ion is required. It is very, very, very important to have the calcium, right? It is very important to have the calcium at that point of time, right? So what is going to happen when there is the change in the membrane permeability? The calcium channel, they will open, and obviously if calcium channels will get open, there will be the influx of calcium, right? Here you are not going to say that calcium will come out from endoplasmic reticulum. No, here the calcium channel will open and obviously there will be the influx of calcium. Are you getting it? Yes or no? Are you getting it? And even bicarbonate ion is equally important here. Done, bache? Done, bache? Yes or no? Yes or no? So calcium channels open, calcium enters sperm. Okay, calcium enters sperm. Clear? This is important. Calcium is going to enter the sperm here. Is that clear? Yes or no? Guys, be quick. Calcium will enter here. Now, the next point is, right, remember in the chapter chemical control and coordination, we talk about some second messengers. We discuss some second messengers there. Right? We discuss some second messengers there. Anyone? Some second messengers we used to discuss there when we talk about the hormonal control. We used to say, now hormones are first messengers, the hormones that bind to the surface of the cell, right? Then inside they will initiate a cascade, right? They will initiate a cascade. Basically, a multi-step process will take place within a cell. Right? So some second messengers are required. Like, let's say you are not allowed to go somewhere, right? You need to pass a message there, right? Let's say there is one government office, you are not allowed directly. So obviously you will pass the message to the guard there that please go inside and tell them that this is my concern, allow me to come in, this and that. Same is the case when you talk about the hormones. Let's say there is a cell, right? Cell is having the cell membrane. So the hormone, some hormones, their receptors are present on the cell membrane. So when that hormone and their receptors, they join now within the cell, some cascade, you can say that a cascade will start. Basically a multi-step process will start. So after binding of that hormone to receptor within the cell, you will see some second messengers. Okay, you will find some second messengers there, right? So basically it is the kind of signaling molecule there, right? So I'm going to relate this part with that portion so that you can understand the application of that part. So after the calcium enters the sperm, what is going to happen? This calcium and bicarbonate, right, they will initiate that cascade, that they will initiate some, you know, multi-step processes there, right? Right, it will initiate that multi-step process there. Are you getting it? It is going to initiate that multi-step process there. And then what is going to happen? What is going to happen, bache? That cyclic, remember cyclin adenosine monophosphate, that CAMP forms. Remember this? Yes or no? I'm not saying that you have to remember it for the NEET examination, but at least you should know the application there. So CMP production will be there, CMP form, and then again the events of acrosome reaction will occur, right? Obviously after this topic we will talk about the acrosome reaction, right? We have to talk about the acrosome reaction, but before that you should know the entire picture. So now you know that there are the egg membranes. Now you know the sperm will get charged. Now you know that sperm is going to bind with that egg, and then further the steps will take place. So any doubt up to this part? Any doubt? Yes, bache. So first of all, these proteins will be removed so that some receptors which are present, right, the receptors which are present, they will get exposed. Moreover, the pH increase will be there, the permeability will change, the calcium ions will open, the calcium influx will be there in the sperm, and that calcium and bicarbonate, they will initiate a signaling process within the cyclic adenosine monophosphate, CAMP will form within the sperm, and this is going to result in the acrosome reaction. It will help in the initiation of that acrosome reaction, which is very important for the sperm binding. That is why charging is important. Okay, that is why the charging of the sperm is important. Any doubt, bache? Yes. That is why the charging of the sperm is important here. Clear, bache? Clear, bache? Yes or no? Yes or no? There are some other processes also, but as of now, tell me, any doubt? Any doubt? Sure? Sure? So egg is basically the oocyte plus egg membranes. The oocyte plus egg membranes. Clear, bache? Done? Okay. So now let's talk about the steps of this, the steps of fertilization basically. Okay, okay, clear, sure. So the very first thing that you need to know is, so basically there are 5 steps, right? Basically there are 5 steps when we talk about this fertilization. The first step is, can you tell me about the first step? The first step is chemoattraction, right? Chemical attraction. Chemoattraction of what? Chemoattraction of sperm to the egg. Okay, chemoattraction of sperm to the egg. That is the first step. Okay, that is the first step. So do you know about the anti-fertilizants and fertilizants? Do you know about the anti-fertilizant and fertilizant? Have you ever heard about it, bache? Yes, anti-fertilizant and fertilizant. Anyone? Yes or no? And tell me, you were aware of these points or not? Have you ever studied it in this way? See, again, I'm telling you, we have to master the NCERT. We have to revise it from NCERT, but the topic clarity should be there, right? If you understand the topic, if you know that proper flow, so obviously reading NCERT will be interesting. You'll be able to make the notes in an easy way. You'll be able to revise it in an easy way, right? So that is why it is important to understand all the aspects of a topic. Okay, and we have sufficient time as of now. I have shared one strategy video with you all now, that how to score the 360 out of 360 in biology. Do check that video. So I have given you the syllabus bifurcation there as well. So if you guys will follow it, right, you will be able to cover your syllabus on time, and then you can revise it as well. Clear, bache? So see the first point, chemo attraction of sperm to egg. This is the first step. So if you remember anti-fertilizant, these are some chemicals, you know, these are some receptors which will help in that binding. So anti-fertilization, fertilizant is on sperm and fertilizant is on that ovum. Right, this is how they are going to bind. So obviously, if that sperm from vagina, from uterus, if it is moving towards the fallopian tube, there are some chemicals which are released, right? There is that chemo attraction there, right? There is that chemo attraction there. Are you getting it? Are you getting it? Yes or no? There is that chemo attraction there. So first point clear, bache? First point clear. Now the second step is, do you know about the second step? The second step is, right, bache? So the second step is binding of sperm to that zona pellucida. Second step is what? Binding of sperm to zona pellucida. I'll tell you how. To zona pellucida. You must be thinking, what about the cumulus? What about the corona radiata? I'll tell you. Then the third step is the acrosomal reaction. Right, acrosomal reaction. The acrosomal content will be released. Right, the acrosomal content will be released. Then sperm will pass through. Right, the sperm will pass through that extracellular envelope. Right, the sperm will pass through the extracellular envelope, and the last step here is fusion of, right, fusion of sperm and egg membrane. Fusion of sperm and egg membrane. These are the steps, right? This is how there will be the recognition of the sperm and the egg, right? There will be the recognition of the sperm and the egg. Are you getting it? Yes, bache. So sperm attraction will be there, of course. It will be there with the help of these chemicals. Yes or no? It is going to help. These chemicals are basically going to help there, right, bache? So this is the first step. So now, what is going to happen here, bache? The capacitated sperm, the charged sperm, the capacitated sperm, it release hyaluronidase. Even this capacitated sperm, na, it releases hyaluronidase. Right, Shamsheer? It is going to release what? It is going to release the hyaluronidase. So it will dissolve the cementing material. Right? Bache, do you know about the hyaluronic acid? Yes or no? Do you know about the hyaluronic acid? I hope you all have heard about it. Hyaluronic acid. What is it? It is hyaluronic acid. It is the cementing material. In the chapter structural organization also we discuss about it. So basically these cells are connected together, right? Because we are a multicellular body. So these are cells are connected together with the help of this hyaluronic acid, right? With the help of this hyaluronic acid. So obviously this hyaluronic acid is like a cementing material. It is connecting all these cells together, and you know, it is very good for our skin, this hyaluronic acid, right? Right, even in some shampoos you might have heard about it, that it is Tresemme ad is there, right? That it is having the hyaluronic acid. So basically it is good for our skin. It is good for that rejuvenation of our skin as well. It is good for hair as well, Clear, bache? So when a sperm is charged, that is why the charging of sperm is important. So when a sperm is charged, when it is capacitated, right, hyaluronidase, which will dissolve hyaluronic acid, right? It is going to dissolve hyaluronic acid. So capacitated sperm will release hyaluronidase, which is going to dissolve the cementing material hyaluronic acid. So now imagine that oocyte which is covered with egg membrane. So that hyaluronidase will help in passing that cumulus, will help in passing that corona radiate, and then the sperm, with the help of some receptors, it is going to bind with the zona pellucida. Is that clear? It is going to bind with the zona pellucida. I'm repeating this point again. Zona pellucida is acellular. This layer is made up of glycoproteins. Right? It is a previous year question. It is made up of glycoproteins. So here you will see some zona pellucida 1, zona pellucida 2, zona pellucida 3. These are the receptors which are present on this zona pellucida. Clear? The sperm is going to bind here, right? These are basically some proteins to which the sperms are going to bind. Are you getting it? So zona pellucida 1, zona pellucida 2, zona pellucida 3. These are the proteins to which the sperms will bind. Is that clear? Is that clear? Yes or no? Right? So zona pellucida will bind to the sperm, and then after it the acrosomal reaction will start. Clear, bache? Clear, bache? Yes or no? Clear, clear, clear? Done? Yes or no? Right? So these are the glycoproteins. These are the glycoproteins which are present on this zona pellucida. So first of all, that sperm. Now you know that the sperm is charged. Let's say, imagine the structure of sperm is like this. Imagine it's like this. Right? So here, you know, some proteins are present. So these proteins present on the sperm structure, first of all, they will bind with. First of all, it will bind with this ZP3. Right? It is going to bind with this ZP3 protein. Right, bache? Right? So when it comes to the primary binding of sperm to zona pellucida, it is possible with the help of this ZP3 receptor. Right? So this ZP3 will bind with the, it will bind to sperm receptors. Clear, bache? It will bind to sperm receptors. So this is the primary binding. So just imagine. Okay. So just imagine that's the sperm structure. It is having some receptors, right? And this is that ovum. This is the zona pellucida. So they are going to bind together, right? This is ZP3. So this is the primary binding. Clear, bache? This is the primary binding. This is how it is going to bind. So now when they'll bind, what will happen? The acrosomal reaction will be initiated. This is what you need to remember, right? So after this primary binding, when there is this receptor interaction, right, when sperm protein, when it will bind to this ZP3, right, when it will bind to this, right, ZP3, then what is going to happen, bache? Yes, calcium ions are already present. Calcium channels will open. Right? Calcium channels will open. Yes or no? See, what is going to happen? Here, the calcium channels open. Okay. So, influx of calcium will be there. Influx of calcium will be there. So, because of that, what is going to happen? The acrosomal content will release. Do you know about this acrosomal content? Acrosomal content, it releases. Okay, acrosomal content, it releases. Do you know about this acrosome? Yes or no? Do you know about this acrosome? Yes or no? You know that acrosome, it is present here in the head region of the sperm. So this acrosome is fully loaded. It is fully loaded with what? It is having sperm license. It is having sperm license. It is having that zona license. It is having that hyaluronidase. It is even having that corona radiata dissolving enzymes, right? So it is fully loaded with the content that helps in the fertilization, that helps in dissolving the membrane. And this acrosome is formed by Golgi apparatus, right? So Golgi apparatus is the organelle that used to form this acrosome. Clear, bache? It used to form this acrosome, right, bache? Done. Clear. So during this, so when this ZP3 and the sperm receptor, when they will bind, when this cross-linking is there, so calcium channels open, right? In the sperm, there will be the influx of calcium, and that will result in the release of acrosomal content. So now obviously, bache, sperm is also having, right? Sperm is also having the egg membrane, of course, isn't it? The sperm is even having the acrosome, yes or no? Yes or no? And this egg is having this zona pellucida. So of course, let's say this is how they are binding. Just imagine this is how they are binding. So when this acrosomal content will be released now, bache, so this sperm egg membrane here, it will, shed off some part of this. See, the content is getting released here. This content is getting released here. Okay. So what will happen here, bache? The part of plasma membrane shed off. The part of plasma membrane shed off. I'm not saying that entire plasma membrane of sperm will shed off. No, not at all. It's just that this acrosomal content will be released here. It will dissolve the covering, right? It is going to dissolve the covering here. And see, sperm is still, and even sperm need that. Sperm should remain bound to that egg itself, right? So only the part of plasma membrane will shed off. Only some part of it will shed off, and the acrosomal content will be released. And see here, you will see some acrosomal process. An acrosomal process will form here. And you know what will you see here? Actin protein. Remember, actin protein. If you have attended my cell cycle and cell division ka one shot, we were talking about the cleavage furrow, right? When we talk about the cytokinesis in the animal cell, we talk about the cleavage furrow, and even in cytoskeletal element, we talk about the actin filaments. These are the microfilaments. So these acrosomal process, here it is basically the actin. Here it is what? It is basically the actin. So sperm bonded with zona pellucida. The primary binding is with the ZP3 receptor, right? So when the acrosomal content will be released a bit, obviously only a part of plasma membrane will shed off. Other receptors now they will play their role. ZP2, ZP1, now they are going to play their role. Clear, bache? So sperm will bonded with the zona pellucida by other ZP2 receptor interaction. Clear, bache? Yes or no? Done. So now, as I said, the third step. Now the sperm content will help in dissolving the zona pellucida, yes or no? This acrosomal content will help in dissolving the zona pellucida, yes or no? Now what is going to be the third step here, right? What is going to be here? Now the sperm and the egg plasma membrane fuses, right? Sperm and the ovum plasma membrane fuses after it. Clear, bache? They both will fuse together. Right? So if they will fuse together, because we are done with the zona pellucida. Zona pellucida with the help of zona license, right? Because acrosomal content is released. So obviously, and even there will be the formation of that acrosomal process also. So obviously the zona pellucida will be dissolved. Now the next step is sperm and the egg plasma membrane will also fuse. You know na, egg is also having plasma membrane, sperm is also having plasma membrane. Now this is going to fuse. So basically I am explaining you the acrosomal reaction here. Clear, bache? Done. So when the acrosomal content will be released, obviously a part of this membrane will shed off. Now the zona pellucida will be dissolved, and finally the sperm and the egg plasma membrane will fuses to each other. Is that clear? Yes or no? Hanji bache, is that clear? Right? So obviously the sperm content, the next step will be the sperm content will be passed to the egg. Okay, the sperm content will be passed to the egg. So see, all these steps are justified. That first of all, there will be the chemo attraction, anti-fertilization, fertilizing on sperms, fertilizing on ovum, binding of sperm to zona pellucida with the help of that ZP3 receptors, and then the acrosomal content will be released, zona pellucida will be dissolved. Still the sperm and egg, they are still bonded to each other, right bache, right bache. And then what will happen? The sperm will pass through these perivitelline space, through this extracellular envelope, and finally the fusion of sperm and egg membrane will be there. Any doubt? Yes, any doubt? So see, capacitation, the capacitated sperm here. So in epididymis, right, this is the sperm, this is the seminal vesicle. So when it is ejaculated, you guys can see that sperm structure is covered with so many proteins and carbohydrates. Now in the female tract, right, these unnecessarily, these proteins and carbohydrates that are covering it, they will be removed and the receptors will be exposed. Clear? This is the capacitation. This is the capacitation. So plasma membrane of epididymal sperm contains a complement of surface molecules, right bache? And here, when sperms are exposed to the female tract environment, seminal plasma coatings along with some of the surface molecules will be removed, exposing the portions of molecule that can bind to zona pellucida of the oocyte. So I hope it is clear to you. Clear bache? Now the next is this. See the acrosomal reaction, right? The acrosomal reaction. Clear bache? Clear? The acrosomal reaction. So 100 of sperms attracted to the corona radiata begin to break through the barriers of granulosa cell and they will approach the zona pellucida. First step clear? They are going to approach the zona pellucida. So contact with the zona pellucida triggers the acrosome reaction, causing sperm to release the digestive enzymes. They will break down the glycoprotein membrane of zona pellucida, helps to expose the oocytes. Plasma membrane. So here you people can see. So see, when the first of all, you know bache, mostly when we Google this process, right? So they also show us some images. That images are of sea urchin, right? That images are of sea urchin. Like whenever you will find this word, na, jelly coat, jelly coat. So remember, it is not a mammalian egg. It is not a mammalian egg. It is actually the sea urchin. Right? You are not talking about the humans basically. Yes, you are not talking about the humans basically. You are talking about the sea urchin, right? So basically, when we study this process in the developmental biology, they have studied the sea urchin. They have studied other amphibians, right, bache? Right, bache? So in the mammals, you are not going to see this jelly coat. This jelly coat is considered as tertiary egg membrane. So in the case of humans, you are not going to find it, right? You are not going to find it. So in our case, these steps of fertilization are also different. In the case of this organism having jelly coat, the acrosomal reaction will occur first and then there is the binding with the zona pellucida. But in our case, what is the scenario? First of all, there will be the binding to the zona pellucida, and after that binding, the acrosomal content will be released. So see, this content will help in dissolving the egg membrane. So finally, the sperm and the plasma egg membrane and the oocyte egg membrane will fuse. This nucleus, the sperm nucleus will be passed here, bache, right, and then see here, some other processes will also occur. That is the cortical reaction. So tell me, any doubt? Any doubt? We'll explain the cortical reaction, but any doubt here? Yes? Any doubt? If there is any, do let me know. This topic is very interesting, isn't it? This topic is actually very interesting. Done, bache? Clear? So next is prevention of polyspermy. Bache, you know that in our case, what is the scenario? One egg will fuse with one ovum, isn't it? One egg will fuse with one ovum. So you know that the sperm is also having 23 chromosome. The oocyte is also having 23 chromosome, right? Together when they will fuse and we will get the zygote, we will get the 46 chromosomes. So in the nature, we go for the monospermy. Monospermy is preferred. Why? Because if there will be the polyspermy, the number of chromosome will get disturbed and it can result in the formation of a new species, right? If we want to attain the if we want humans, so obviously the chromosome number should be 46, otherwise it can result in abnormalities, it can result in the formation of another species, right? So the monospermy is preferred in nature, right? Monospermy is preferred in nature. The concept of twins is totally different. Okay, the concept of twins, it is totally different, right? In the case of twins, sometimes, you know, after the formation of zygote, when there is the embryonic other developmental stages, so sometimes at that time, you know, the initial blastomeres, they get separated and they grow into that they grow into that monozygotic twins wala part, right? And sometimes, you know, there are two different eggs and two different sperms, they fuses them, then you will have obviously dizygotic twins. Two zygote will form here from one zygote. Here from one zygote we used to get the twins. So that concept is totally different, but the polyspermy is not preferred in the nature. So when it comes to the block to polyspermy, so obviously there is fast block and there is the slow block as well, right? There are two blocks basically. One is the fast block and second is the slow block, right? One is the fast block and second is the slow block. Fast block is like, you know, the sperms, when they fertilizes the egg membrane, at that time there is a change in the permeability, there is a change in the ionic concentration. So when there is change in the ionic concentration, right, let's say initially, right, when the egg membrane, it was not fused, the membrane potential was -70 MB, but later on, because there is the calcium influx also, it will become 20 plus 20 MB. So in that case, right, in that case the other sperms cannot fuse it. Clear bache? Other sperms cannot fuse it, right? So how are we going to study it, right? So first of all, when acrosomal released, yes, so in the mammals we don't talk about the fast block much, we discuss about the slow block of polyspermy. And when it comes to the prevention of polyspermy, yes, as per your NCERT, there are two reactions. One is the acrosomal reaction and another is the cortical reaction. So together they prevent the polyspermy, right? When you talk about the slow block to polyspermy, you talk about fast block means immediately change in membrane potential. You will see it in amphibians, right? Change in membrane potential, right? So membrane potential will change. So obviously the sperm is now, the egg is now not ready to bind with any other sperm. It's a fast block, but it will be for the shorter duration. Now the next is the slow block to the polyspermy, where you will have acrosomal reaction and the cortical reaction will be studied. Now you know about the acrosomal reaction, isn't it? Yes, Bache Jyoti. Now you know about the acrosomal reaction, isn't it? So acrosomal released enzymes, right? Remember this, acrosomal released enzymes, they modify the zona pellucida. Do you know that? Acrosomal released enzymes, they modify the zona pellucida receptors. Okay, so such changes will take place in the case of mammals. What is going to happen in the case of mammals? The acrosomal released enzymes, right? Acrosomal released enzyme, they modify the zona pellucida receptors. They modified the zona pellucida receptors, like they will modify zona pellucida sperm receptors that it cannot bind with any other sperm. They modified the zona pellucida sperm receptors that they cannot bind to other sperm. They cannot bind to another sperm. Are you getting it? That is why we even consider this acrosomal reaction as a slow block to polyspermy. So this is the very first step, Jyoti. This is the very first step, that acrosomal released enzymes modify the zona pellucida sperm receptor that they cannot bind to the another sperm. Clear, bache? So now next step is, so after all this binding, right? After all this binding, and let me tell you, the nucleus here in the case of ovum is germinal vesicle. If you will ever find this word, germinal vesicle, so the nucleus of this ovum is known as germinal vesicle. So after this acrosomal content, right? See, this is zona pellucida, This is the perivitelline space. And here you have this plasma membrane of ovum, right? Here you have this plasma membrane of ovum. Now, bache, below this plasma membrane, you find some granules. Even they are made up of Golgi apparatus, right? Even they are made up of Golgi apparatus. These granules are cortical granules. What are they? They are cortical granules. As I said, they are also made up of Golgi apparatus. So now you know that acrosome has modified the sperm receptor, and during this binding, when there is the sperm and egg plasma membrane fusion, when that sperm content will be passed here to the egg. So from endoplasmic reticulum, listen to me, in the case of sperm, I was talking about the calcium channels. Okay, I was talking about the calcium channels, but here what am I saying? That here in the case of this oocyte, here in the case of this egg, this endoplasmic reticulum is there. So after this binding from this endoplasmic reticulum, the calcium ions will be released, right? So intracellular calcium, you know na, endoplasmic reticulum is a storehouse of calcium. Endoplasmic reticulum, smooth endoplasmic reticulum basically. Let me be more specific. Smooth endoplasmic reticulum, it is the storehouse of calcium ions. So after these reactions, from the smooth endoplasmic reticulum, the calcium ions will be released. So when there will be the release of these calcium ions, it will make these cortical granules to fuse with the plasma membrane. Okay, because of these calcium ions, what is going to happen, Subhasini? These cortical granules, they will fuse with the egg membrane, right? They will fuse with the egg membrane, and because of that, this content of cortical granules will be released here in this perivitelline space. The content of this cortical granules will be released here in this perivitelline space. Okay, so this cortical granule is having nothing, it is also having that enzymes. Okay, it is also having this that enzymes. Clear, bache? Clear. So after this release, what is going to happen? The cortical granules, when they will released here in perivitelline space, now the egg will become a structure which cannot further bind to any of the sperm. Right. So cortical granule, there is the exocytosis. Exo means outside. So exocytosis of cortical granules will occur. Right, exocytosis of cortical granules will occur, and it is calcium mediated process. So it will occur with the help of calcium. So that is why we also say that it's a calcium mediated process. So any doubt here, bache? Yes, any doubt here? It's a calcium mediated process. Clear? This is even comes under the activation of egg also. See, look at these steps. So sperm will come, sperm makes contact with egg, right? So in the case of mammals, this is the second step. Acrosome reacts with zona pellucida is not the second step. In the case of mammals, right? In the case of mammals, the sperm is going to bind with the zona pellucida, then acrosome. Okay. Yes. In the case of mammals, the sperm will bind to the zona pellucida. Then there is the release of this acrosome, right, bache? Right, acrosomal content will be released. Plasma membrane of sperm and egg will be finally fused. Sperm nucleus will enters the egg here. The cortical granule will fuse with the plasma membrane. Right, in this perivitelline space, they will make this thick coat. Further, there will be no binding of another sperm to this ovum. Clear? Done? So look at this diagram. Just look at this diagram, quick. Clear? Sure? Done? So see, this is the growth of acrosomal process. Again, we studied in the case of that sea urchin. See how after the release of that acrosomal content, this acrosomal process, which is made up of actin, it will be formed, right? Further, see this sperm nucleus, how it will be passed here. And then there is the, after this step, there is the exocytosis of cortical granules. And as I said, it's a calcium mediated process, right? So entry of sperm nucleus will be there finally. Clear, bache? So finally, the fertilization membrane will form like this. And now no other sperm can penetrate the ovum. Is that clear? Yes, any doubt? Is that clear, bache? Is that clear? Yes or no? Because you know that after it there will be the formation of zygote. After it, what is going to happen? The zygote formation will be there, and that mitotic arrest will overcome now. Sure? Sure? Okay. Clear? Done? So, yes, the next step. Now, when bache this sperm, right? You know that basically in the case of a zygote, the more cytoplasmic content is from the egg itself, right? It's the mother's. Just a minute. Yashoda ma'am sent top 100 MCQs of living word. Yashoda, first of all, attend this lecture. I have already sent it. First of all, attend this lecture, revise this, right? Focus here, then I'll send it. Okay? That. So now you know that sperm will pass its nucleus. Basically, it is the pronucleus, and along with that, sperm will also pass the centriole. Do you remember? Sperm is having two centriole, distal and the proximal. Distal and the proximal. That one centriole in the case of sperm will form that flagella. It will form that exonym, the rod-like structure, 9+2 arrangement of flagella. And this proximal centriole, it will be passed to the egg. Right? You know what? This oocyte, it is without centriole. This is important. Mammalian egg, mammalian oocyte, it is, right? Mammalian egg, mammalian oocyte, it is what? It is without, right? It is without the centriole. So no centriole means no formation of spindle apparatus, means metaphase 2 arrest will be there for sure, right? So now when the sperm will fuse, it will pass the proximal centriole. It is going to pass the proximal centriole. So obviously in the egg, in that egg, then metaphasic arrest, right? It will overcome. It will overcome. So basically after fertilization, there will be the completion of meiosis 2 in the case of females. Right? In the case of females. So obviously at this time, what is going to happen? You know that you will be having that proper nucleus now. So basically now this egg, we used to call it as ooted, and one more polar body will be released. Right? One more polar body will be released. So after this fertilization, when the sperm nucleus, when the sperm centriole will be passed, so this centriole will overcome. Right, this centriole will help in overcoming that metaphase 2 arrest. Right, it will form that spindle apparatus. Of course, the process will be completed. Now this egg is also known as ooted, and the polar body will be released. Polar body will be released. Now that nucleus of that egg is good, right? Now it will bind with the sperm nucleus. It will bind with the nucleus of that spermatozoa. Clear? Clear? So after that, what will you get? You will get the zygote. You will get a diploid zygote, right? And zygote is what? It is a totipotent cell, right, bache? It's a totipotent cell. It is having the ability. It is having the ability to. It is having the ability to grow into a complete organism. Clear, bache? Clear, bache? So now this is what we need to study. This is what we need to focus. Clear? So this process, like, it is taking place, this fertilization is taking place in ampulla. I told you already, the widest part of the fallopian tube ampulla, where the fertilization will take place. Now after it, further cleavage will be there. Remember, give, sip, go. After fertilization, there is the cleavage. Fertilization will form the zygote. Zygote will undergo the cleavage. So now what is going to happen? The cleavage will occur. So obviously the cleavage will initiate in the cleavage will initiate here in the fallopian tube, right? This cleavage will initiate there in the fallopian tube. So as I said, zygote is what? Zygote is a totipotent cell. So bache, now we will talk about the cleavage. Right, we will talk about the cleavage. So cleavage is also considered as rapid mitotic division. What is it? It is also considered as rapid mitotic division. And let me tell you, I'm not talking about the humans only, right? But mammalian cleavage is slowest in the animal kingdom, right? Mammalian cleavage is slowest in the animal kingdom. Remember this. Mammalian cleavage is slowest in the animal kingdom. So after 30 hours of formation of this zygote, the first division will occur and we will get two cells, two blastomeres. What are we going to get? We are going to get two blastomeres. Are you getting it? Two blastomeres. Are you getting it? Are you getting it? So I am explaining it here. Cleavage, right? Cleavage. So as I said, mammalian cleavage is slowest. Mammalian cleavage is slowest in animal kingdom. Clear? So in the cleavage, right? But what is the, why do we say that cleavage and mitosis, it is different? Why do we say that cleavage and mitosis is different? No doubt in some books, you know that it is written like this, that cleavage is the rapid mitotic division, right? That cleavage is the rapid mitotic division, but it is different from, as I said, it is different from the mitosis. Why? Because in the case of cleavage, G1 phase and your G2 phase, they are absent. Do you know about G1 phase and G2 phase? They are absent. That is why it is rapid. Okay, that is why it is rapid. G1 phase and the G2 phase is different, right? When we talk about the cell cycle, na bache, so G1 phase is the growth phase. It is the post-mitotic phase. It is the pre-DNA synthetic phase. So in G1 phase, there will be the protein synthesis. There will be the formation of RNA, right? So cytoplasmic growth is there. What is there? The cytoplasmic growth is there. Right? What is there, bache? The cytoplasmic growth is there. Now, I told you that your zygote will undergo cleavage first and it is going to form two blastomeres. That is what I just said, right? It is going to form two blastomeres. That is what I just said, right? So, it will undergo cleavage. So, there will be no G1 phase. There will be no G1 phase. There will be no G1. No G2 means cytoplasmic growth will not be there initially. So when the division is cleavage, there is no cytoplasmic growth. When the division is cleavage, there is no cytoplasmic growth. Clear, bache? Clear, bache? So no cytoplasmic growth means see, this is the zygote after, and it is still covered with zona pellucida. Let me tell you, it is still covered with zona pellucida. So after division, it will form two blastomeres, but size will not increase. After another division, it will form four blastomeres, but again the size will not increase. So for initial, right, for the initial stages, when there is the cleavage, the size of the zygote will not increase. Are you getting it? The size of the zygote will not increase. Cytoplasmic growth means increase in size, but there will be no increase in size here. There will be no increase in size here. And as I said, still, just remember it. It is covered with the zona pellucida. It is still covered with that zona pellucida. Right? Here also it is covered with that zona pellucida. It is covered with that zona pellucida. Clear, bache? Clear? This is about the cleavage. Now, another important point here in the cleavage is right, this is the first step. After 30 hours, there will be the division. Now see, normally, you know what happened? Initially, the zygotic genes. Obviously, we have the nucleus here. We'll be having the genes here. We have the nucleus here. We'll be having the genes here. So that genes used to form the protein, and that is going to take over the function of that cell, right? Right, that will help in the functioning of that cell. But actually, it is not the case. For initial developments, for initial cleavage, for initial stages, like let's say up to 13th cleavage, right, the zygotic genes are not doing anything. The maternal genes, right, the maternal proteins, because we know that zygote, the majority of cytoplasm is from the mother itself, from the ovum itself. So maternal genes used to do the work, right? Maternal proteins used to take care of everything. But in the mammalian cleavage is again an exception. From the starting itself, the zygotic genes, right, they are transcribed, they are taking the charge. Okay, they are transcribed, they are taking the charge, right? This is what you need to remember. And in this cleavage is basically, see, first of all, human egg is alecithal. Do you know that? Human egg is alecithal. Alecithal means no yolk or very less amount of yolk. Either you can say no yolk or very less amount of yolk is there, right? So it is alecithal. Mammalian egg is what? It is alecithal. Without yolk or very less amount of yolk is there. So when it comes to the cleavage, this cleavage here in the case of mammal is holoblastic. Holo means total cleavage is there. It is holoblastic. It is holoblastic. Total cleavage is there. Like no yolk is there. So obviously properly all the divisions will occur, first point. Now this cleavage is also known as rotational. Right? It is also known as rotational cleavage. Do you know why? Do you know why I'm telling you about the mammalian cleavage? It is holoblastic, it is rotational, and it is asynchronous. It is asynchronous. Right? It is asynchronous. Now, what is the meaning of rotational? Now, you know it's a alecithal without yolk. So, all the blastomeres will divide here. All the blastomeres will divide here, first point. Second is, it is rotational. What is the meaning of rotational cleavage? See, first of all, I told you it is slowest. So, after the formation of, after 30 hours of zygote formation, there will be the first division. Right, Shriharsha? After 30 hours of zygote formation, there will be the first division. So actually, why do we say it is rotational? Because the first plane, right of division, right, this is meridional. This is meridional. So that is how the blastomeres will get divided. Right, one student is asking the difference between blastomere. Blastomeres are the cells which will form after this cleavage, right? So first division is going to be meridional, and when you talk about the second division, it will be equatorial. It will be equatorial. So you see that this division is not all same, right? So it looks like so rotations are there, na? First division is like this, second division is like this. Okay, so rotational division is there. So this cleavage is rotational. So first is meridional, the first is meridional, and the second is what? The second is what? Second is equatorial, right? The second is what? Second is equatorial. So when you talk about the mammals, right, it is going to be the alecithal egg, holoblastic cleavage will be there, right? Holoblastic cleavage will be there, and it is going to be rotational. It is going to be rotational. Clear, bache? So there is the unique orientation of blastomeres to each other. Okay, there is the unique orientation of blastomeres to each other. First is meridional, second is the equatorial, right? So I'm also using the word asynchronous. It means all the blastomeres, they do not divide at the same time. Right? They will not divide equally. They will not divide at same time. They will not divide equally. Clear, bache? Done? So as I said, the zygotic genes will take charge from the starting itself. Okay. So mammalian genome is activated during early, right? During early cleavage. This is what you need to take care. So understood why are we saying rotational cleavage and what is the meaning of asynchronous cleavage? Clear? And the next point is, bache, the unique property of compaction that we will see here in the case of mammalian cleavage, right? In the case of mammals, this is the unique property that you people are going to see. Do you know about the compaction? Do you have any idea what this compaction is? Yes, bacho. Do you have any idea what this compaction is? It is again a unique property in the case of mammals. So you know that after one division, 2 blastomeres will be there. After second division, there will be the 4 blastomeres. After third division, there will be the 8 blastomeres. Right? Let's say this is the case. After third cleavage, there will be the 8 blastomeres. So there is one study, you know, in the case of, obviously in the case of rat, basically after third cleavage, yeah, you can say that up to third cleavage, up to third cleavage, what is going to happen? If you will see that blastomeres are having some space in between, right? Let's say after third cleavage, total 8 cells will be there, na? Let's say that is the scenario. Obviously, it is covered with the zona pellucida. That is not the case. It is covered with the zona pellucida. That is not the case, right? But you will see that up to third cleavage, right? The blastomeres, they have some spaces in between. Blastomeres have space in between, right? But after third cleavage, what is going to happen? All these cells, they will come closer because some proteins will get released, some cadrins will get released. है ना? Then they will become compact. They will become compact. So initially up to third cleavage, in the case of mammalian cleavage, you will see the space in between. So after third cleavage, the compaction will start. The blastomeres will come closer. The tight junctions, right? The tight junctions will form in between. So the compaction will occur, right? The compaction will occur. Is that clear? Is that clear? Yes or no? Yes or no? This is what now we are discussing about the embryonic stages. Okay, so after 30 or after the 30 hours after the formation of zygote, we will have the 2 cell stage, 2 blastomeres are there, then 4 blastomeres, then 8 blastomere stage. So you will see this property here, you will see this compaction here. Now bache, 8 celled, right? Now you know that it is the embryo formation. So 8 celled embryo will divide, you know it, right? 8 celled embryo will divide, will form 16 celled embryo, and that is what? That is morula. Remember, that is morula. Yes or no? What is it? It is morula. Yes, Suhashini. Subhashini, what is it? It is morula. Okay, it is morula, 16 celled stage embryo. Right, it is 16 celled stage embryo. So it looks like mulberry, like, you know, ball of, it looks like the ball of blastomere, 16 cell. And then it will divide, right? It will divide. Now you will see the growth as well. There will be the 32 celled embryo, and that is what? That is your blastula. That is your blastula. So that 32 celled stage will form what? It will form your 64 celled blastocyst, right? It will form your 64 celled blastocyst, right? So morula, blastula, blastocyst. Now what is it? See, blastocyst. This diagram is important. Huh? Now here your cells will, the cells here, they are going to arrange themselves. Okay, the cells here, they will arrange themselves here. The structure is blastocyst, na? This is the structure basically, blastocyst. So after 32 cell stage, we have this 64 cell stage, and that is the blastocyst. Now, what is blastocyst? Cyst. From the C, you have to remember one word, that is seal, means cavity. So here in blastocyst, you will see blastocele, you will see the cavity. And here from this blastula, you will see that, you know, these cells will start arranging themselves properly. Now see the layer in the surrounding, this layer is the trophoblast cells, right? Trophoblast layer, trophoblast cells, and of course it is nutritive. It is nutritive. It is nutritive. And here this cell mass is ICM. It is inner cell mass, which will form the embryonic structure. Okay, this is the inner cell mass, and this cell mass is going to form the future embryo. It will form the embryonic structures. Are you getting it? It is going to form the embryonic structure. So this is blastocyst. So mass of cell is ICM, inner cell mass. It will form the future embryo, and it will also form extra embryonic layers. So this trophoblast, it is nutritive, and it will even form bache chorion. It will even form extra embryonic membrane chorion. It is going to form this chorion. Now this blastocyst is still covered with zona pellucida, right? There is no hatching of this blastocyst from zona pellucida yet, right? It is still covered with this zona pellucida. Up to this part, all clear. These are the embryonic stages, bache. In next 30 to 40 minutes, we'll finish it. Okay, so up to this part, all clear, yes or no? Up to this part, all clear. 8 cell, 16 cell, 32 cell, 64 cell, and then this is the part. This is the part. Now see, come to this diagram. Okay, see this. Zygote, 2 cell, 4 cell, 8 cell, 16 cell, and finally this blastocyst will reach here. So you can say that after 7 days of fertilization, after 7 days of fertilization, implantation occurs. After 7 days of fertilization, implantation occurs. That is, what is implantation? The fixing of blastocyst to the endometrium lining of the uterus. The fixing of this blastocyst to the endometrium lining of the uterus. This is the implantation, and it will take place after 7 days of fertilization. So actually what is happening, bache? This blastocyst takes, this is very interesting. Blastocysts take near about 5 days to reach uterus. Do you know that? It is taking near about 5 days to reach uterus. You know, now parallelly some other changes are also going on in female's body, right? Do you know that? Parallelly some other changes are also going on in female's body. You know that after, see, we are saying after 7 days of fertilization, implantation will occur. And as I said here, this blastocyst will take, or you can say that this zygote will take near about 5 days to reach the uterus. So 5 days after release of egg means in the female body, luteal phase is going on. This is what you guys can conclude, right? So in the female body, can we not say that there is the luteal phase which is going on, the secretory phase which is going on? So at this point of time, you know that in female body, progesterone is present, isn't it? Progesterone is present at that time. Yes or no? Yes or no? Related with the menstrual cycle, na? Related with the menstrual cycle. This is what happening here, na? So we are saying after 7 days of fertilization, implantation will occur. So after, let's say egg released today, 7 days after that egg release, again in the female body, luteal phase is going on. In the female body, secretory phase is going on. In the female body, corpus luteum is there, which is releasing the progesterone, and progesterone is anti-abortion hormone. Progesterone is the pregnancy hormone. Progesterone is acting on the endometrial lining here. It is helping in the growth of that endometrial lining. So no doubt this process is taking place parallelly, simultaneously. Our endometrial lining is also growing, isn't it? Our endometrial lining is also growing. Yes or no? Yes or no? Yes or no? So basically that progesterone, you know that even, so you know that parallelly progesterone is also there in our body, even some amount of estrogen is there. So it is helping in the growth of that endometrial lining, right? Done, bache? It is making it compatible for the implantation. So meanwhile, what is happening here? We need to show, implantation will be later. Meanwhile, what is happening here? Here, the endometrial cells, they are growing, they are proliferating, bache, they will become swollen. You know, they will become swollen. They are loaded with the food now, right? They are loaded with the reserve food now. You know that? Right? The endometrial lining, you know that it is the innermost lining where the implantation occurs. Are you getting my point? Endometrial lining, right? This endometrial lining is, this endometrial is the innermost layer where you know that implantation will occur. This is important. It's a PYQ, right? Implantation occurs here. Implantation occurs here, isn't it? Implantation occurs here. Clear, bache? Clear, bache? So endometrial lining, it is dividing, it is proliferating, it is acquiring that reserved food. And now, you know, we also use the word decidua for it. right? Endometrial lining is having different names, you know, we call about strata decidua, basal decidua. I will not go in the depth of it, right? So I'm just using the word decidua as of now, right? I'm just using the word decidua. Is that clear? Is that clear? Yes or no? Is that clear? Yes or no? Still, if there is any doubt, do let me know, bache. Right? Do let me know if there is any doubt. Clear? So this is decidua. Clear? Clear? Because now the cells are swollen. Cells are swollen. Progesterone is there. Progesterone is helping in it. And you know what, bache? Initially, when this growth is going on, no? When this growth is going on, obviously because there is no cytoplasmic increase, so nucleocytoplasmic ratio, right? Initially, so there is no cytoplasmic growth, size will not increase, the zygote size will not increase, but later on, near about after 13th cleavage, right, you will see the G1 phase and the G2 phase will be added, and now there is the cytoplasmic growth. So obviously, initially what is happening? Your nucleocytoplasmic ratio is increasing. Nucleocytoplasmic ratio is increasing initially, isn't it? Isn't it? When there is the cleavage going on, nucleocytoplasmic ratio is increasing, and you will see here, you will see during this growth that the cell size will reduce. The cell size will reduce, new cells will form, and they will be smaller, right? Zygote is having larger size, and then further multicellular structure will form, so that cells will have smaller size. But overall, all in all, that multicellular structure is not growing, it is not increasing its size, but you will see it will be divided into small blastomeres. Clear, bache? Clear, bache? So initially this is the process. Clear? So as I said, then now this is totally the endometrial lining, the cells are fully loaded with the reserve food material, and you know, they even become glandular, right? We know now progesterone is acting on the endometrial lining, it is increasing the size, it is making them swollen, and even it will make them glandular also. Glandular means secretory. Secretory means obviously it will release some nutritive things. Now, this is what you need to understand. You know what, when after this fertilization, no doubt this blastocyst will take near about, you can say that blastocyst in 5 days will reach here in uterus. You know, in 5 days it will reach here in uterus. After fertilization, blastocyst will take 5 days to reach here in uterus. Now you know that no doubt it is covered with a zona pellucida, but don't you think it doesn't have anything from where it can take the nutrition, right? It doesn't have anything from where it can take the nourishment, isn't it? So this blastocyst, it is basically taking the nourishment from the uterine milk. Actually, this endometrial lining is secretory now. It is releasing something. We used to call it as uterine milk, right? We used to call it as uterine milk. So this blastocyst is taking its nourishment from this uterine milk, which is released by endometrium. And then what is going to happen, bache? Right. So this blastocyst will take near about, right? This blastocyst will take near about 1.5 days to get implanted here in endometrium. Are you getting it? It will take near about, yes, 1.5 days. First of all, it will take 5 days to reach here in uterus, and till that it is taking nourishment from the uterine milk, from the secretions of endometrium. Now after reaching here, it will take another 1.5 days, and it will get implanted here in endometrial lining. So that is why in totality we used to say that it takes 7 days, right? After 7 days of fertilization, implantation occurs, right? After 7 days of fertilization, implantation occurs. See now, 5 days to reach here in uterus, 1.5 days for that implantation. So in totality 7 days are there, isn't it? In totality 7 days are there. Yes or no? Yes or no? So slowly this blastocyst will get implanted here. Now you have to understand this part, right? You have to understand this part. So you know that even the trophoblast is here. And here you have the inner cell mass. Okay, so now this trophoblast, it will start releasing proteolytic enzymes. This trophoblast, it will start releasing proteolytic enzymes. What will it release? It will start releasing proteolytic enzyme. Proteolytic. It will cause the breakdown of proteins, right? It will cause the breakdown of protein. So these proteolytic enzymes, you know, they will even digest this zona pellucida. They will even digest this zona pellucida. So when these proteolytic enzymes will be released now, so there will be the hatching of this. There will be the hatching of this egg, hatching of this, you know, this embryo from the zona pellucida. So when it will reach here, when it will reach here, so obviously there will be the hatching. There will be the hatching of this egg from the zona pellucida, and slowly these proteolytic enzymes, they will create a space here, right? Like just imagine, Shamsheer, it is more like that. Like let's say you are digging the ground, right? Then you will stay there in that space, and again someone is covering you with the soil again. It's more like that. Like see, the proteolytic enzymes from the trophoblast will be released, right? So that proteolytic enzyme will help in hatching of this embryo from this zona pellucida. Moreover, they will create a space here in this trophoblast. They will create a space here in this endometrium. Not in trophoblast, in endometrium, right? And slowly endometrium lining will again cover this blastocyst. This is the story. Okay, so these proteolytic enzymes will help in that process also, right? So once it will get implanted here, now the placenta formation will start, right? So initially it is just taking the nutrition from the endometrial secretions, but now the placenta formation will start. Is that clear? Until then, pregnancy will be maintained by your progesterone. Now I'll tell you what is going to happen next. So up to this point, if there is any doubt, do let me know. It's very interesting. Trust me. Up to this point, up to this part, do you want anything? Do you want me to explain anything? Yes? Done? Sure? Sure? Tell me, any doubt? Sure? Sure? Are you sure? Okay, so do you know why this blastocyst is still covered with zona pellucida? Why this blastocyst is still covered with zona pellucida? Yes? Why this blastocyst is still covered with zona pellucida? Do you have any idea? To avoid the ectopic pregnancy, right? To avoid what? To avoid the ectopic pregnancy. So now what is the meaning of this line? What is this ectopic pregnancy, bache? What is the meaning of this ectopic pregnancy? See, implantation should be there in the endometrial lining. Implantation should be there in the endometrial lining. If this blastocyst, if this embryo, if this zygote, it gets implanted in any part other than endometrial lining, let's say in the ovary, let's say in the fallopian tube, it is the ectopic pregnancy, right? See, you know that endometrium, this uterus, this endometrium, it is meant for this implantation, right? So obviously it can help in the nourishment of that zygote. It can help, it can support that growth. But let's say if the implantation occurs in ovary, obviously that particular embryo will not survive. It can even damage the ovary. So if it will get implanted in fallopian tube, it can even destroy the fallopian tube. So it can result in worse conditions. So just to avoid the ectopic pregnancy means when implantation is not there in the endometrial lining, it is any place other than, if the implantation is there at any place other than this endometrium, it is known as the ectopic pregnancy. Is that clear, Bharat? Is that clear? So that's the point here. Clear, bache? That's the point here. So that is why it is still covered with the zona pellucida. So as I said, the enzymes will be released. They will, create a space there in the endometrial lining, right? And then that endometrial lining will be covered. And see, this is how the implantation will occur, right? Up to this part, all clear. Now, once this blastocyst get implanted here, now you know that you have the trophoblast, you have the inner cell mass here, right? You know about these things, isn't it? You people know about these things, isn't it? Isn't it? Now what's the next part? What's the next part here? This trophoblast, it is going to form one extra embryonic layer, that is chorion, right? See, when you talk about the germ layers, you know that humans are triploblastic. Germ layer is your ectoderm, middle one is your mesoderm, inner one is your endoderm. Clear? These are the germ layers. So they are also known as embryonic layers. We have one more word, that is extra embryonic layers, right? Extra embryonic layers or extra embryonic membranes. So they are not going to make the embryo, but they will help in that, their nourishment and their growth. So extra embryonic membranes, we have amnion, the chorion, the yolk sac, and the allantois, right? And the allantois. These are the extra embryonic membranes. Amnion, chorion, yolk sac, allantois. Amnion, chorion, yolk sac, allantois. But this amnion, you know that baby is going to grow in the uterus. Within the uterus, that baby will grow. So obviously, you know that amniotic cavity is there, right? The amnion is lining that amniotic cavity. So you know that here we have that fluid also, right? Amniotic fluid is also present there. Remember, amniotic fluid is also present there, will act as a shock absorbent, will help in the growth. That's the point. Amnion. Now this chorion will help in the formation of placenta. Chorion is that part of the embryo, which will take part in the placenta formation. You know that placenta, temporary endocrine gland, very important for the growth and the nourishment, right? Now when you talk about the yolk sac and the allantois, honestly the function of allantois is not so clear, but allantois, it is believed that when that embryo is growing, allantois help in that excretion, right? Yolk sac, you know that initially it will help in that RBC formation and all, right? From the yolk sac, basically, you know that precursor germ cells will also form. So that is how these extra embryonic membranes, they help in the growth, right? That is how these extra embryonic membranes, they help in the growth. Is that clear, bache? Is that clear? So just after fertilization, this is how the process, yes, this is how it will proceed, right? This is how it will proceed. So after fertilization in human, you can say that after 7th day, there will be this implantation. So after 12 days of fertilization, Jyotika, after 12 days of fertilization, this blastocyst will be totally buried here. It will be totally buried here in this endometrium. It will be totally buried here in this endometrium after the 12 days of fertilization. Is that clear? After the 12 days of fertilization, it will be totally buried here. Clear? Clear, bache? Yes, is that clear? Yes or no? Is that clear? Okay, sure. So yolk sac, you know that, right? So I told you the function of yolk sac, I told you the function of this allantois as well. So up to this part, all clear? Fine. Okay, so now when this implantation will occur, obviously it is going to take time. I'm not saying that it will occur in a day. No, as I said, after 12 days of fertilization, this blastocyst will be totally buried here in this endometrium. Okay, and slowly this trophoblast will start the growth. This trophoblast will start forming some, finger-like structures. It will start forming some finger-like structures. Now, after implantation, placenta is going to be there, right? So these finger-like structures are nothing. This is the chorion extra embryonic membrane. It is the part of embryo which takes part in the placenta formation. Now, what is this placenta? Placenta is a kind of intimate connection. It's not a direct connection, let me tell you, right? It's not like that directly mother supply or the fetus supply, fetus blood supply will get mixed. No, No, let's say, right, from the mother's side, this endometrial lining, this decidua part, this endometrial lining, this decidua part will, you know, it will form some finger-like structure. From this baby's side, bache, you need to understand the process. So please listen to me very carefully. So from that baby's side, there will be the formation of chorion, right? So placenta will form. In this way, the placenta will form, and this placenta is nothing. It is the intimate connection. It is the intimate connection, right, bache? And basically what is going to happen, from mother's blood supply with the help of diffusion, the baby is going to get the nourishment, right? The baby is going to get the nourishment. So this is how it proceeds, right? This is how it works. Are you getting my point? This is how it works. Is that clear? Yes or no? Is that clear? Yes or no? Done, bache? Is that clear? Yes or no? Right? So that's how the further growth will be there. Just look at this. Just look at this amazing picture, bache. This is the embryo. So you know that initially when you compare the vertebrate embryos, they are quite same. Later on, we'll, in late pregnancy, we'll see that how we'll find the special characters will arise. Then we will see that, okay, that's the human embryo, right? Remember this evolution from evolution, that biogenetic law or born wear law, that's initially there is the formation of the, general character, and then there is the growth of, there is the formation of the specific characters. I hope you remember. And see, this is how it will go. Fertilization, cleavage, then compaction. Now you know the meaning of compaction. So after compaction, slowly differentiation will occur. Now these cells, they will start arranging themselves. You know that the cavity, this blastocyst will form. So after it will hatch from that zona, right? So after hatching from zona pellucida, implantation is going to occur. This is the uterine epithelium. This is the proteolytic enzyme. You will see slowly how cells, the trophoblast cells are penetrating, right? They will deeply bury themselves here. So just look at this. Just look at this diagram, bache. This is very important. This is the uterine lining, right? So near to the uterine lining, you will see this trophoblast layer, right? That is how it works. That is how it works. Clear, bache? And slowly you will see the growth here, right? This inner cell mass will also become differentiated. It will also form epiblast, hypoblast, and slowly other layers will form. We will talk about this. So finally you will see a structure like this, where that fetus that is totally covered with the amniotic cavity is there in the surrounding, right? You will see other extraembryonic membrane structures. So see, this chorion is forming these finger-like structures. Are you getting it? Yes or no? Yes or no? Wait, I have another beautiful diagrams as well. Clear? So that is how it will go. So see, this epiblast, hypoblast, we'll talk about these layers, but as of now, if there is any doubt, do let me know. So finally, this baby is there in the mother's womb, right? This baby is there in the mother's womb. So it is present within this amniotic cavity. It is covered with that amniotic fluid, right? It will covered in that amniotic fluid. And look at this umbilical cord. It is right? This umbilical cord is attached to our navel. It is attached to our navel here. So this point is showing the connection in between the mother and the baby, right? We have that belly button now, that is navel basically. So that is a connection. It is the part to which the umbilical cord is attached. So umbilical cord is showing that connection. That placenta is forming that cord-like structure now. It is. So that is how that baby is present, right, in the uterus. So it is the uterus which is the womb. So now just look at this connection first, right? Look at this connection first, and then one by one we will talk about the other steps also. You see here, can you see this? Can you see this? Right, this yolk sac, the placenta, right? So this is the maternal blood supply, right? This is the uterine artery and vein. This is the chorionic villi, right? So in the case of placenta, placenta is hemochorial because we have that blood connection, no intimate connection is there. So in our case, this is the hemochorial placenta. What is it? It is the hemochorial placenta. And bache, that yes, that intimate blood connection is there. And from mother side, that is decidua of that endometrial lining, and these are the chorionic villi. These are the chorionic villi. This is something important, okay? This is something important. So just mark it. So chorionic villi and uterine tissue, they will become interdigited with each other and they will form this placenta. right? So they will form this placenta. Clear bache? Clear bache? So on the basis of intimacy, let me write it here. On the basis of intimacy, right? On the basis of intimacy, it is the deciduate placenta. It is the deciduate placenta. Right? What is the meaning of this deciduate placenta? It means, right. So here there is a close connection in between the chorionic wall and the uterine wall. That is the endometrial lining. You can see it here. You can see it here. The close connection is there. Right? The close connection is there. So that is why we call it as deciduate placenta. Clear, bache? Clear, bache? Okay, so even this chorion, it penetrates deeply into the uterus. Yes, bache Bharat, this chorion, it deeply penetrates the uterus. So we also call it as intimate placenta. Oh, sorry, interstitial placenta. Right? Why? Because chorionic villi, they deeply, okay, chorionic villi, they deeply penetrate uterine tissue. They deeply penetrate uterine tissue. That is why we call them as interstitial placenta. And third word is, bache, discoidal placenta. Right? Third word is discoidal placenta. Right? Do you know the meaning of this discoidal placenta? This word is very common. Discoidal placenta. It is on the basis of distribution of villi. On the basis of distribution of villi. Actually, no doubt, I'm telling you now that chorion will form the villi. So imagine a circular structure. So only at one point in a disc-like area, you will see the finger-like projections. Right? Right? Like let's say this is the structure. So only at one point, you people will see the disc-like, right? Like this, finger-like structures will be there. Right? Only at one place you will see the distribution of these villi, these chorionic villi. Clear, bache? Right? So whole of the chorionic surface is covered with the villi, but the villi later, they will be there in a disc-like region. Right? Chorionic villi present in the disc-like structure. Right? So it is discoidal placenta. Okay, it is discoidal placenta, and on the basis of histology, because there are the human placenta is interdigitated, right, bache? So there is the deep intimate connections basically, right? This you know, the blood supply are so close, so we even call it as, when we study this, we even call it as hemochorial placenta. What type of placenta it is? It is the hemochorial placenta. Clear, bache? Hemochorial placenta. So discoidal, because this is present at a one place only. Wait, I'll show you the diagram if I have. So see, this is the baby, this is the fetus, right? So this is the amniotic sac in which this baby is present, and here we have that placenta. Here we have this placenta. Just look at it. The diagrams here are very important, bache, right? They are very beautiful and they are very important to understand a topic. Clear? They are very important to understand a topic. Clear, bache? Clear? And see this deep intimate connection. Look at it. This is the, so see, from mother, the blood supply, see, have a look, right? So see this intimate connection, this umbilical arteries, right? So it is attached to that fetus. This is going to that fetus. So here you have the arteries and the veins. So mature chorionic villi, which is covered with syncytiotropoblast. We will see that trophoblast will further grow, right? It is divided into different layers, but we don't get the question from that part. So we will limit up to the NCERT only, right? So here you guys can see these intimate connections, right? So this is some space in between. Intervillous space filled with maternal blood, mature chorionic villi. This is the chorionic plate. So that is how the intimate connection is there. So that is how it is the hemochorial placenta, because of it. So basically the diffusion will occur, right? Maternal blood supply, fetus blood supply. So diffusion will occur. So directly things will not pass here and there, right? It is the diffusion which is going to occur, right? It is a diffusion which is going to occur, right, bache? Clear, clear, clear. So this placenta will provide the nourishment, will help in the removal of excretory waste, also will protect the baby from the birth defects. I hope you know about the word teratogens, right? They cause the birth defects. It can include anything, the chemicals, the physical factors, right, that used to causes the birth defect. So placenta also protect the baby from these birth defects from the teratogens. So that is how it is present. Clear, bache? That is how it is present. See, done? Any doubt? Yes, bache. Do you have any doubt here? See, these diagrams are important. Okay. So I hope you are getting the idea that how it is present. See, placental septum, intervillous space, right? The other layers, the maternal blood vessels. This is how they are present. Clear? Clear, clear, clear. So fetal section, middle section, maternal section is there. And here you can see the amnion and the chorion as well. Okay. Done, bache? Done. So now when this placenta will start forming, na, after the formation of this placenta, now, so now you know that the placenta formation is there, right? So this implantation, now it indicates the pregnancy, right? It indicates the pregnancy, right? At this time, what will happen, bache? One hormone will be released, that is human chorionic gonadotrophin, right? This hormone will be released, that is human chorionic gonadotrophin, right? Human chorionic gonadotrophin, HCG. Right? It is HCG, human chorionic gonadotropin. So when there is the placenta formation, it is the hormone which is released by placenta. And it is a question, you know, that the hormones released by placenta. So your placenta will release what? Your placenta is going to release the human chorionic gonadotropin. And this is the hormone which indicates pregnancy. Okay, it is the hormone that indicates pregnancy, right? So when females, you know, they check the pregnancy, when they do the pregnancy test, you know that they with the help of that Prega News and all, they can check it now. They need to put the drop of urine there, and even the blood test is there. So basically they used to check this particular hormone, human chorionic gonadotropin, right? So obviously it is formed only in the case of female. It is not there in the case of male, because males will not get pregnant. We have the only the one pregnant male is the hippocampus. Okay, so basically what is there? What is that? The HCG will form. So it is the HCG, human chorionic gonadotropin, that indicates the pregnancy. Now, when HCG will start forming, so obviously with the help of blood, right, it will even pass a signal to that corpus luteum. Okay, you know that if there is no fertilization, the corpus luteum get degenerated. You know it or not? You know it or not? When there is no fertilization, you know that the corpus luteum will get degenerated. The corpus luteum will get degenerated. We know about it very well, isn't it? We know about it very well. So now here what is going to happen, bache? Right. So corpus luteum, right? So when there is the formation of HCG, human chorionic gonadotropin, it is going to send a signal to the ovary. With the help of blood, right, it will pass to the ovaries. So corpus luteum will get to know that, okay, pregnancy is there. So now the corpus luteum will not get degenerated. The progesterone will still be there. Okay, the progesterone is still present there, because initially the placenta is not, right? Initially the placenta is not, you know, the placenta is not that, it has started forming only, right? So obviously initially the corpus luteum will maintain the pregnancy. Or let me tell you, in first trimester, or yes, in first trimester, the presence of corpus luteum in ovary should be there, at least for the first month of pregnancy, right? At least for the first yes, Yes. So initially at least for the first month of pregnancy, the presence of corpus luteum should be there. Even after the formation of placenta, the corpus luteum should be there for some time. If corpus luteum is removed from the ovaries, there will be no pregnancy. Right? There will be no pregnancies, and slowly, the placenta will take over the function. Okay? The placenta will take over the function. Is that clear? Is that clear? Done. So HCG is going to pass a signal to corpus luteum. Corpus luteum will keep releasing progesterone and estrogen, which will maintain endometrial lining, which will act on mammary glands for the lactation, which will prepare the mother body for the pregnancy. Clear, bache? Right? Which will prepare the mother body for the pregnancy. So that is how this placenta is going to help. And slowly, you know that this placenta will also release another hormones. Right? It will also release that somatotrophins. Right, it is also going to release what? It is also going to release the somatotrophins, right? Somatotrophins, human chorionic somatotrophins, and it is also known as human chorionic lactogens. Actually, bache, this hormone na, honestly, initially its role was not clear, right? Human chorionic lactogen. Basically, it is human placental lactogen. Initially, the function of this particular hormone was not clear, right? It was believed in other animals. It was believed that it is acting on the mammary glands. It is helping in the milk production. It was believed, but later on, you know, we see this hormone is having the different role, right? I'll give you one example here. Now you know that we humans, right, we babies, when we grow within our mother's uterus, we are totally dependent on our mother. I'm not talking about right now I'm telling you about the functions of placenta, and then we'll come back to that part, you know, inner cell mass and all. Whatever is important for you, we are going to discuss it, and maximum by 11:40 I'll end this session. So as of now, you please listen to me. So you know that we babies, we are growing within our mother's uterus. We are going to take all our nourishment from our mother. Okay, this placenta is helping in that. With the help of that placenta, all the essential things we are going to get, plus it will also help in the removal of waste. So when we grow, right, RBCs will also form. We are growing basically. So you know that after 21 days of that fertilization, even the heart, a heart-like structure, not properly a heart, but the pumping organ will start forming. The blood supply in our body will start after 21 days of fertilization, like in first month of the pregnancy, right? In the first month of the pregnancy, after the 21st day, we'll see the heart-like structure will form and the pumping of blood will start. That is why, right, during initial stages of pregnancy, doctors used to check they used to perform the USG, they used to check the heartbeat. So if heartbeat is there, like baby is fine, baby is growing, like this, right? Later on, they go for another things, the sonography and all. This is how it works. So this is what you need to remember, that after 21 days of that fertilization and all, what is going to happen? The heart-like structure will form, right? Usually we say that 5th week of pregnancy, we roughly consider it as 5th week of pregnancy also. So what I want to, heartbeat is going to form. So what I want to mention here actually, so we are dependent on our mother. We are going to take all our nourishment from our mother. So our body, see, we are just like a parasite on our mother's body, isn't it? Don't you think that the babies that grow within the mother's body, they are just like the parasite on mother's body? The only difference is that we don't belong to the same species. We belong to the same species. Like in the parasitism, this is what we used to say now, one is host, another is the parasite, and one is the host, another is the parasite, right? They are from different species, but in our case, we belong to the same species, and mother wants to carry the baby. But in one way, you can say that we are just parasites on our mother's body. Even after our birth, we are totally dependent on our mother, and till today, even I am too dependent on my mother. Okay? So it's like that. So basically, the body, the mechanism is in this way. See, we have that survival instincts, right? It is in our system. It is coded in our system. We have that survival instincts. So the hormones, this human chorionic somatotrophins, you know, these hormones, they even make they even help in, you know, taking the glucose at a faster rate than the mother's body. Like we can utilize more and more glucose from our mother's blood. We can derive more and more glucose from our mother's blood. This hormone, this human chorionic somatotropin, can also help in that way, right? And even, you know, we can utilize our hemoglobin, that baby's hemoglobin, right? You know, adult hemoglobin and fetal hemoglobin is little different. It is little different. Fetal hemoglobin is more efficient, right? Its affinity to oxygen is more, right? Even if there is less number of RBCs, still that fetal hemoglobin can bind to the oxygen very quickly. Its affinity is more. So these hormones, they maintain it, right? So with the help of human chorionic somatotropin, we can even take the glucose from the mother's blood at a faster rate in comparison to the mother's body. So we take everything from our mother's body for our personal growth, right? And when that baby within the uterus, when feels like now the space is not good for me, I want to go out, right? Now I cannot grow here, I cannot be dependent here. So obviously that fetus will initiate fetal ejection reflex as well, right? Fetal rejection reflex as well, right? So when you talk about the placenta, you talk about the human chorionic gonadotropin, which indicates pregnancy, right? And then the human chorionic somatotropin, human placental lactogen, which will also help in milk production. With the help of this hormone, we can even take out, you know, more and more glucose at a faster rate from mother's blood, and even the oxygen, right, at a faster rate. Okay. And then this placenta also release the corticotropin hormone. Right? The corticotropin hormone is also released from this placenta, and that establishes the timing of birth. Right? It establishes the birth timing. Corticotropin hormone, it establishes the birth timing. Clear, bache? Clear, bache? And even this helps in increasing the secretion of what? Cortisols. So even help increased secretion of cortisol. Right? Right? So that's how it works. And later on, even this placenta will also release the, but this is how it is going to work. Clear, bache? It will also release the relaxant, but later. Done? So this is how it works. Clear, bache? Clear? So placentation is there. Now placenta is providing the nourishment. The intimate connection is there. So baby is getting everything. So now you know the next step, isn't it? Now you know the next step, isn't it? You know the next step. Implantation, give, sip, go. So gastrulation will occur, right? So first of all, understand here. I told you that we have, in that blastocyst, we have inner cell mass, right? And then here in the surrounding, we have the trophoblast cells. You know about it, yes or no? Yes, bache, do you know about it? In the blastocyst, this is what I just said, that here we have the trophoblast. Here, this is the ICM, inner cell mass, and the cells of trophoblast that are in contact, right? The cells of trophoblast that are in contact with this inner cell mass, we also call them as cells of Robber, right? They are also known as cells of Robber, right? So no doubt this trophoblast is forming the chorionic villi, and all the processes are going on here, right? This is what we know. Now this inner cell mass, you know what, bache? This inner cell mass will also grow. It will also divide. It will also arrange itself, and it will form two structures. It will form epiblast and hypoblast, right? It is going to form two layers. This inner cell mass, this ICM, it is the future. It is the future embryo. This ICM is what? It is the future embryo. So it is going to form the bilaminar disc. What will it do? It will form the bilaminar disc, right? It will form two layers. One is the epiblast, another is your hypoblast, right? One is your epiblast, another is your hypoblast. Clear, bache? Clear, bache? One is what? It is your epiblast, another is your hypoblast. So this is the bilaminar disc. Two layers will form, right? Two layers will form. Basically, you will see here the cell there, right? you will see that how yes, now these cells will form the structures basically. Like if you talk about the epiblast, overall no doubt inner cell mass is going to form your embryo, and this when it comes to the trophoblast will just form only one extra embryonic layer, that is chorion, right? So now this epiblast, it is the one which will form embryo, it is the one that, yes, it is the one that will form embryo, right bache, and it is the one that will even form the amnion, right? It is the epiblast which is going to form the embryonic body and the amnion. Embryo and the amnion will be formed by this epiblast, and this hypoblast will form your yolk sac. It is the one that will form that yellowish yolk sac. Clear bache? Clear bache? So this question can be asked in the NEET examination, that which part is going to form the embryo? This is the ICM, inner cell mass, and this bilaminar disc, epiblast, embryo, and the amnion, hypoblast will form the yolk sac. So there will be the formation of, you know, the germ layers now, right? After this step there is the gastrulation. Now after implantation, gastrulation will be there. So gastrulation means the germ layers will form, right? The germ layers will form. And how that germ layers will form, bache? They will form with the help of some morphogenetic movements, right? Some morphogenetic movements will occur, like slowly, some type of movements like epiboli, this boli, that boli. There is no need to go in that depth. So obviously you have some cells. These cells will start foldings. Some infoldings will occur. Sometimes invaginations will occur. Sometimes infoldings are in this way that it can, you know, some layers will get separated and you will see the new layers. So no doubt we have no need to understand this process in detail. Ultimately, you know that in gastrulation there is the formation of germ layers. Gastrula formation is there. So these are the morphogenetic movements. These are the morphogenetic movements which will result in the formation of gastrula. And once these germ layers form, they will form different organs, right? Because after gastrulation we have the organogenesis. So that is how it works. So this part here is important. Clear bache? This part here is important, and let me show you the layers now. So if you will see this, like slowly, you know, firstly your endoderm will form, right? Your mesoderm is the middle one, ectoderm will form. So that is how you will see, right? That is how you will see that slowly these layers will form, right? And they will form the different structures. Let me show you the, okay, yes. So now can you see this here, bache? Can you see here this inner cell mass? This inner cell mass here is forming two different colored structure. So this one is your epiblast, right? This one is your hypoblast. Can you see this? Slowly see how hypoblast is growing in this way and epiblast is growing in this way, right? And you can even see the days also. You can even see the days also. So this your epiblast, right? Epiblast is growing in this way, that is, it is forming, it is covering, it is forming the amnion. Can you see this? Your epiblast is growing in this way, that it is forming this cavity like structure. You know that it is the amniotic cavity, isn't it? You all know that it is the amniotic cavity, and here within the embryo will grow, right? And when you talk about this hypoblast, it is forming this primitive yolk cavity. Now just remember, always remember, inner cell mass will form bilaminar disc. Bi means two, laminar means layer. Two layers will be there. Epiblast, hypoblast. Epiblast will form your embryo. E for epi, E for embryo. It will form your amnion. EA, same kind of sound. It will form amnion. Hypoblast will form your, because hypoblast, in its spelling we have HYPO, right? Hypoblast. So Y means yolk sac, so it will form the yolk sac. That is how you can remember. And now see, primitive streaks and all the structures are forming. These layers will also split. You will see some mesodermal cells in between. Even this is also, you know, in the surrounding, it is also forming the structures. So that is how you have to remember. That is how the further growth of layers will occur, right? Right, that is how the further growth is taking place. Clear bache? Clear bache? And here in the NCERT the diagram is given like this. So see, the blastocyst will get implanted. So this diagram is important, right? There was so one PYQ was there. In that PYQ they were asking what is A, what is B, what is C, what is D, what is E. So diagram is important here. Okay, this diagram is important here. Is that clear? Yes or no? Is that clear? हाँ, the same type of stages are here. ठीक है बच्चे? Okay. So epiblast, hypoblast. So remember, this trophoblast is what? This trophoblast is multipotent, that it cannot form all the structure, it will just form chorion. And when you talk about this inner mass, it is your pluripotent. Actually, it is developing into, you know, the complete structure. Okay? Okay? Done? And now these germ layers, bache, see, this is the fate of germ layers. So your ectoderm, the outermost germ layer, so it will form your, you know, CNS, central nervous system, your hair, the retina, the epidermis, the skin, the mammary glands, they are formed by ectoderm. See, outer surface, central nervous system, neural crest, right? Mesoderm, the middle layer. Your skeletal system is mesodermal in origin. Your heart is made up of mesoderm. Your muscles are mesodermal in origin. Endoderm will form your digestive tube, the pharynx, the respiratory tube. Right. So this is what you need to remember. So zygote, blastula, gastrula. Gastrula is having the germ layers formed by the morphogenetic movements, and these germ layers will form different organs. So it is important to remember. Okay, it is important to remember. So you guys can see here. So epidermis of skin, right, epithelial lining of mouth and rectum, sense receptors, cornea, lens, nervous system, adrenal medulla, right, tooth enamel, they all are formed with the ectoderm. And endoderm is forming the liver, the pancreas, the thymus, the thyroid, epithelial linings, bache, lining of urethra, urinary bladder, right? And here your mesoderm is forming, see, adrenal cortex, lining of body cavity, notochord, skeletal system, muscular system, excretory system, right, bache, reproductive system except germ cells. Okay? Okay, so this is what you need to remember. This is important. And here you can see the development. You know that in the case of humans, the pregnancy is for 9 months. Okay, so in 9 months, 0 to 3 months, first trimester, 4th to 6th month, second trimester, right, 7th to 9th month, third trimester. So first trimester, 12 weeks, where, you know, abortion is easy and it is legalized. If a female need MTP, medical termination of pregnancy, she can go for this, because initially the skeleton is, you know, cartilaginous, it is not bony. So here, you know, another 20 weeks, 24 weeks. So here 36 weeks, or sometimes it can be 38, 36 or 38 we consider. It can vary. So first trimester is of 3 months, right? So this is the first trimester. So in first trimester, you know that heart will form. Heart will basically form in the first month of pregnancy or in the 5th week. This is what you guys can consider. This is the second trimester, right? This one is the third trimester. So see, initially week 4, heart begins to beat. Arm buds appear, liver, pancreas and gallbladder start to form, spleen appears. So bache, this is theoretical. This is what you need to remember. I'll provide you the PDF. You guys can revise it from there. Okay, so second में week 5 में eyes start to form, leg buds appear, hand appears as pedals, blood begins to circulate, facial features start to develop. This is the weeks, not the trimester. 4th week, right? See, initial 12 weeks are first trimester. So here we are talking about the first trimester. See, the major development is here in this first trimester. Week 6, lung starts to form. Actually, your respiratory system, it forms at the end. Why? In the end, basically. Why? Because we are getting the, you know, all that gaseous exchange from the placenta itself. Week 7, hair follicles start to form, elbows and toes, they are visible. 8th week, face begins to look human-like, external ears start to form. So that is how you need, that is how the human baby develops. Clear, bache? Clear? Done? So now let's revise it from NCERT, and then firstly let's talk about the parturition and the lactation, and then we'll revise it from the NCERT. Clear, bache? Then NCERT reading will be there. Okay? Fine. So now you know that when that fetus, when that baby is growing within the mother's body, obviously, right? Now that space is not sufficient enough. So fetal ejection reflex will start. Fetal ejection reflex will start. Fetus is going to start it, right? That now throw me out. I don't want to live here. Throw me out. And you know that, right? So basically the setup is in this way. In the surrounding, you know that myometrium, perimetrium is there in the uterus. So first of all, fetal ejection reflex will start, and because of that, in the case of female body, right bache, from the posterior lobe of pituitary. I hope you remember it, right? From the posterior lobe of pituitary, posterior lobe bache, right? You know na, posterior lobe is not the true endocrine part. When you talk about this endocrine system, you know that we have hypothalamus. Now that hypothalamus is forming, it is attached to the pituitary through the stalk-like structure, right? We have the anterior lobe of pituitary. We have the anterior adenohypophysis having anterior and the middle lobe, and we even have the posterior lobe of pituitary, which is also known as neurohypophysis. Posterior lobe is also known as neurohypophysis. It is also known as pars nervosa. Bache, it is directly under the direct neural regulation of hypothalamus, right? It is under the direct neural regulation of the hypothalamus, right? So hypothalamus is going to make these hormones, will give it to the posterior lobe, will give it to the neurohypophysis. So from here the oxytocin will be released. Along with oxytocin, it is also the ADH will be released, but in pregnancy we need to talk about this hormone, that is oxytocin. So oxytocin is bache your milk ejection hormone. It is also known as milk ejection hormone. It is also known as milk let down hormone, right? It is also known as birth hormone. It is also known as, right, bonding hormone or the love hormone, right? When when there is during this bonding and love, this oxytocin forms, its secretion increases. So it is also known as milk ejection hormone, milk let down hormone, birth hormone. So bache, when fetal ejection reflex will start, so the posterior lobe of the mother will start releasing oxytocin, O for out, O for out. So it will help in the birth. How? This oxytocin this oxytocin will start it will act on smooth muscles. You know in the uterus, in the myometrium, the smooth muscles are present. They are fully grown now. It is the largest smooth muscle, right? In a pregnant female, the uterus smooth muscles are the largest one. Okay, so oxytocin will act on these smooth muscles. So what is going to happen? The rhythmic contraction and relaxation, isn't it? The rhythmic contraction and relaxation, and bache, it is vigorous, right? The rhythmic contraction and relaxation of muscles will be there, but it is vigorous. It's very fast. And sometimes you might have heard about this key in the case of female, the false labor pain starts. The female feels like that is the labor pain. Like when there is a vigorous contraction and relaxation in the uterus, vigorous contraction and relaxation, that we used to call it as labor pain, right? So in first pregnancy, that can even last for hours. You can say that 8 to 12 hours, sometimes 7 to 10 hours. It depends on female's body. In second pregnancy, its duration is short, right? So they are quite vigorous. So it is known as labor pain. And sometimes, you know, that baby is there in that amniotic fluid, in that amniotic cavity. So in the cervix, there is a kind of plug. So when there is the removal of that plug, we also call it as water breakdown, right? Water breakdown in the case of female body. Okay, that amniotic fluid will slowly comes out. It is also a signal now it's the time for the delivery. So this labor pain is quite vigorous. It can last for many hours in first pregnancy. In second pregnancy, it can last for the short duration. So it is because of this oxytocin. So it is also a question, right? So doctors, to induce the labor pain, right, when females, they are in hospital, so they used to give the oxytocin injection. They used to give the oxytocin injection because it is going to initiate that labor pain. It is going to initiate that contractions. Clear, bache? Sometimes female can even face the false labor pains, right? Sometimes, you know, during that growth period, they feel some contractions which can last for a few months, but it is not sure always that it is the labor pain. So doctors, they induce the labor pain by giving the oxytocin. Clear? And this will start the rhythmic contraction and relaxation. So obviously it will be like baby will start coming out from that birth canal, from that cervix and from that vagina. So that is how the oxytocin helps in. And even at that time, the relaxin is also released. So placenta also releases the relaxin, and even it is released from the ovaries as well. Right, placenta also releases the relaxin. This is another protein hormone. So what this relaxin will do, bache, it increases the flexibility of the pubic symphysis. It increases flexibility. It increases flexibility of pubic symphysis. You know na pubic symphysis? Remember pelvic girdle made up of two bones, coxal bones also known as innominates. They are having that pubic symphysis which is linking it, right? Right. So it increases the flexibility of pubic symphysis so that baby can come out, and moreover it dilates the cervix during labor pain. Clear, bache? During labor pain. So that's how it works. Clear? That's how it works. That's how the baby will come out. And meanwhile, this oxytocin, let me tell you. See, bache, from anterior lobe of pituitary, right? From anterior lobe of pituitary, the prolactin will be released. The prolactin will be released. From the posterior lobe, the oxytocin will be released. Now, this prolactin, pro means production. Lactin is related to lactation. So prolactin is responsible for the production, or you can say that secretion of milk from alveoli, right? Secretion of milk from alveoli. Are you getting it? Remember alveoli, mammary lobes, they are having the alveoli. These are the clustered cell. They will release the milk, right? And then it will further pass to the ducts. So production of milk will be initiated by prolactin. It will be initiated by this prolactin. And even progesterone is also going to help there. But that milk ejection, O for out. So when baby will provide the suckling stimulus, milk ejection will be done by oxytocin. So prolactin production of milk from alveoli of that mammary gland, and oxytocin out, milk ejection. So when baby will pass the suckling stimulus from the nipples, from that lactiferous duct, the milk will come out. So this is how it works, right? Right. Thank you so much, Sumo. Okay, bacha? So that is how it works. So this is all about the embryonic development. We have covered it, right? And now let's read the points from NCERT, bache, and then we will end the session. So this is all about the hormonal control. You should write a short note on hormonal control, bache. That is very, very, very important. Clear? Clear? So you know that the zygote will further grow. It will form the morula. Morula will divide and transforms into blastocyst. Very easy it is, right? So now further it will move to the uterus. So blastomere in the blastocyst, they are arranged like this: trophoblast and inner group of cells, which is inner cell mass. Told you already. Then trophoblast layers then gets attached to the endometrium. Trophoblast will get attached to the endometrium, will release proteolytic enzymes, and inner cell mass get differentiated as embryo. So you know that this inner cell mass will form bilaminar disc, and that bilaminar disc includes two layers. One is your hypoblast, another is your epiblast. So epiblast will form that embryo. Epiblast will form that embryo. Clear? So after attachment, the uterine cells divide rapidly, covers the blastocyst. Told you. So blastocysts become embedded in the endometrium, right? This is the implantation and it leads to the pregnancy. Clear, bache? Now you know that after implantation, the placenta will form. So trophoblast will form chorionic villi, which are surrounded by uterine tissue, which are surrounded by maternal blood. Clear, bache? So chorionic villi and the uterine tissue, they will become interdigitated, right? With each other and jointly form a structure which is known as placenta. So placenta is having maternal supplies also, fetal blood supplies also. So it is interdigitated. So it is not directly blood is getting mixed. So with the help of diffusion, the blood will get exchanged. So this is what you need to remember. Thank you so much, Garima bache. No doubt HSP sir is the best. That is why, you know, he is the senior most member of our team, right? Even you are considering me as the second best. Oh my God, I'm so blessed. Thank you. Take it now, please focus here. Don't waste our time and don't waste your time. Okay? So placenta facilitate the supply of oxygen and nutrients. I told you already, right? It also removes CO2. So it is also excretory in function. It is also excretory in function. Clear, bache? So placenta is connected to the embryo through umbilical cord, which helps in the transport of substance to and from the embryo. Clear, bache? So you should know the hormones released by it, So HCG, human placental lactogen, also known as human chorionic somatotropin, even estrogen and progesterone. Right, bache? So in the later phase of pregnancy, a relaxin will also be secreted by placenta. Clear, bache? It will also be secreted by placenta. Clear? Clear? So HCG, HPL and relaxin. It is also a PYQ, bache. HCG, HPL and relaxin. HCG, HPL and relaxin. They are produced in women only during pregnancy. Right? It's important. It is produced only during pregnancy. So in addition, during pregnancy, the level of other hormones, estrogen, progesterone, cortisol, right? Even it helps in that contraction, prolactin, thyroxine, they are also increased. So it is important. Even thyroxine is high. So that's your homework. How thyroxine is helping during pregnancy. This is what you need to tell me. It's a very simple question. How thyroxine is helping, this is what you need to tell me. Clear, bache? Right. So immediately after implantation, the ICM differentiates. The ICM differentiates into an outer layer, ectoderm, inner endoderm, and mesoderm soon appear between this ectoderm and endoderm. Clear, bache? So these three layers will form the tissues, I told you already. So inner cell mass is having the stem cells, which have the potency to give rise to all the structures. So that is why I told you inner cell mass is what? Pluripotent. Okay, it is pluripotent. It can develop into all the tissues and the organs. Clear? Into all the tissues and the organs. Done, bache? Done? So here you guys, so this is your homework, bache. So human pregnancy is for 9 months now. So you have to mention it in the comment section that how many months pregnancy lasts in dogs, elephants and cats. It is important. Okay, that's your homework. You have to mention it in the comment section. Clear? So after 1 month of pregnancy, heart is formed. Important. Highlight it in your NCERT. Right, bache? Right? So by the second month means again first trimester, limbs and digits will form. Limbs and digits will form. Clear? So by the end of first trimester, all the major organ and systems, they are formed. Clear? So limbs, external genitalia are well developed. So first movement and appearance of hair will be there in 5th month. So 5th month means second trimester. Second trimester, right? So by the end of 24th week, the body is covered with fine hair. Fine hair, eyelids are separated, eyelashes are formed. So by the end of 9th month, fetus is fully developed and is ready for the delivery. So you know that the average duration is 9 months, which is the gestation period. So vigorous contraction of uterus will start. And how will it start? Because the fully developed fetus and the placenta will induce uterine contractions, which are the fetal ejection reflexes. So I told you about the hormones as well, and this is what you need to revise. Clear? Clear, bache? This is what you need to revise. So see, suckling stimulus, right? The suckling stimulus will release the oxytocin, which is responsible for the milk ejection. Production is done by your prolactin. Okay, production is done by your prolactin. So I'll share this PDF with you all. You can use it, right? So see, this is how the fetal ejection reflex will start. Mild contractions will start. This is how the baby is going to come out. Right? So onset of labor, then flexion is there, the rotation of head is there, right? The extension, external rotation, and uterus immediately after birth. So this placenta will be removed. Clear? This placenta will be removed. And you know na, elephant, the duration of pregnancy is maximum in the elephant. Elephant is a very slow breeder. It's a very slow breeder. So see, the cervix dilates, amnion ruptures, fetus is expelled, 3rd stage mein the placenta is expelled. So these are the 3 stages of the parturition. So that's all about the human reproduction and the embryonic development, bache. So now what you have to do, I'll share the PDF with you all, but yes, after the class you just need to put a comment. Okay, this is what you have to do. Just write down one comment for this lecture, bache, and I'll share the PDF in the Telegram group. You guys can join our official Telegram group, Unacademy NEET English, and you can even join my personal Telegram group, that is Love Biology by Ambika. All the updates will be provided to you there. And if you have joined the test series, very good. If not, please go and join it, right? Give the test, practice as much questions as you guys can. Okay, Done. So thank you so much. So tomorrow at again at 9 a.m. we have one more lecture, reproductive health in one shot. So reproductive health is a very interesting chapter. We will talk about the importance of reproductive health, about the STIs, and even about the art assisted reproductive technology. Okay. So thank you so much. Bache, the series has started yesterday. Okay. So you guys can still give that test. Today we have the discussion. Fine. So thank you so much. Take care. Bye-bye. Stay tuned and please write one comment.